So, the endocrine system is probably the second most important system in the body behind the nervous system. There's a term called neuroendocrinology, which refers to the study of the interactions between the nervous system and the endocrine system. Basically, how the signals are sent to stimulate hormone release and the gland to produce an increased concentration of a specific chemical. So the purpose of the endocrine system, main purpose is signaling and communication. So it's our main signaling network throughout the body, supports normal homeostatic function. So the endocrine system is made up of a collection of glands that produce hormones. And we can see that picture there. This shows you just all the different glands in the body. And we'll touch on each one of them. But what do these glands regulate? So a lot of them regulate growth and development, also metabolism of macronutrients. Some regulate reproduction. Others will regulate sleep, mood, fluid balance, protein synthesis, and muscle building. So we'll touch on a lot of different glands and hormones in this chapter. So, what are our learning objectives? The first part of this lecture will talk about just like the main function of hormones in the organization of the system. We'll get into different types of hormone action and then also different types of receptors. So, we'll talk about steroid versus peptide receptors and hormones. We'll get into a neat topic called circadian rhythm and how seasonal changes will influence hormone release. We'll discuss the hypothalamus and how it has control over the pituitary gland. We'll talk briefly about anabolic drugs. We'll get into growth hormone, response to exercise, and then there are some few other hormones that aren't listed or topics that aren't listed that we'll discuss as well. Just like the other chapters, we are broken up into 4 different parts. So part 1 discusses different types of hormone, structure synthesis, how they interact with receptors, and then some feedback loops. Let's first talk about the significance of the endocrine system and how it's related to exercise physiology. We want to kind of talk about how this system helps to mediate adaptations to exercise training, basically supports exercise demands as well as recovery. So let's apply hormones to exercise. But before we get into the exercise phys topics, we want to also address the clinical endocrine related topics. So normally I would pause and have the class discuss the difference between clinical endocrine topics versus ex-phys endocrine related topics. So think about it for just a minute. Let's talk about clinical endocrine topics first. So a few clinical endocrine topics. One would be thyroid dysfunction. We'll talk briefly about what hypothyroidism and hyperthyroidism. So that would be kind of more of a clinical topic. I know all of you have heard of diabetes and metabolic syndrome. That has to do with the regulation of insulin, and that's also a clinical topic. Something we won't spend much time talking about is menopause, which occurs in females, and then andropause, which occurs in males. So it's the drop in estrogen or testosterone with age, also clinical. And then hormone deficiencies or overproduction of a hormone. Those are kind of all clinical related topics that we'll briefly discuss, but we'll focus more on the exercise physiology related topics. So what would those be? Anabolic hormones, so anabolic stimulus that results in hypertrophy of muscle, so muscle building. Also catabolic stimuli, so decreased body fat or weight loss we could talk about. We'll talk briefly about steroid use and kind of performance enhancing hormones to basically just improve athletic performance, also physique. So we'll talk about that as well. But yeah, there's a lot more topics that we'll discuss, but the first part of this lecture does kind of go very clinical. We have to understand how is it organized? How does it basically work in order to understand what are the relationships to ex-phys. So let's look at the definition of hormone. So what are hormones? They're substances, they're mainly just chemicals released into the blood from an endocrine gland. So the blood is the transport system. That's how hormones are transported throughout the body. So it's the major transport system for hormones, and it helps them to reach their target cell. Hormones are found in blood plasma. They're found in white blood cells, red blood cells, and platelets. Let's talk about endocrine glands. So what are these, or how are these defined? They're basically just organized group of cells that function as organs to secrete our chemicals or our hormones. We have 3 major hormone types that we will discuss. So keep these hormone types in mind as we go throughout the entire chapter, because every time we learn a new hormone, we will identify what type of hormone it is. So the first type is a steroid hormone. So our steroid hormones are derived from cholesterol. They have the same ring and anatomic numbering system as cholesterol does. Cholesterol is just a lipid. So a lipid is just a broad class of fats. So that's kind of how you want to think of a steroid. But steroid can be used in many different ways. Used for medical treatment, so like anti-inflammatory conditions. They also help to rebuild damaged tissue. They can treat anemia or asthma. Those are called corticosteroids. So there's a difference between corticosteroids, anabolic steroids, and mineral corticosteroids. So basically they exert their action by directly interacting with the DNA of the cell. And we'll talk about that in a second. But anabolic steroids, that's slightly different than our corticosteroids. So these are derived from the male sex hormone testosterone. The anabolic steroids are. They stimulate protein synthesis and hypertrophy. The natural steroid testosterone is important for exercise training response and adaptation. So we will talk about the natural form of testosterone, but also keep in mind there are synthetic forms of testosterone as well. Another class of hormones are called peptides. So peptides made of amino acids. These are very common structures. It's basically 100 amino acids kind of strand together. They exert their signal on a cell by using secondary signaling systems to interact with the DNA. And then next is modified amino acid, or anamines. So anamines, you may have heard me use the word catecholamine before. So these anamines are organic nitrogen-containing molecules. So they're also derived from amino acids, but specifically derived from tyrosine. So I've talked about catecholamines before, epinephrine and norepinephrine. They're also derived from tryptophan, so another amino acid. All right, so let's look at these structures of each one of these. So each hormone type has a specific chemical structure that determines how it's going to interact with the target cell. So how to interact with a tissue or an organ, and that's affected by basically its chemical structure. So let's look at the top. We have our steroid. So one steroid is cortisol. We'll talk about cortisol at the end of this chapter. That's our primary stress hormone in the body, and it's also the primary catabolic hormone in the body. And then one of our catecholamines we see up here to the right is epinephrine. And then you see at the bottom we have insulin, which is a peptide. If you look at the peptide structure, you see it's made of 100 amino acids. If you look closely, you can recognize some of these. So over here we have valine, that's probably glycine, histamine, what's another one? Leucine, tyrosine. So if you know the abbreviation for the amino acids, you can recognize this amino acid strand, or it's when we have multiple amino acids together, it's called a polypeptide. So this is basically just looking at the structure of these hormones. What we also want to learn is our hormone release systems. So hormones have specific release patterns that they undergo, and every hormone has a different one, or one of the three that we see at the bottom. The release of hormones occurs from specific cellular sites in various organs and glands. So synthesized through a series of chemical reactions, then stored and released from the storage sites upon mechanical, neural, or hormonal signals. And those stimulatory signals allow a very specific regulation and control of how much of a hormone is actually released. So let's start with the first type. So there's an endocrine release. So endocrine release secreted directly into the blood. Travels in the bloodstream through the body to reach a target cell. So we look at this picture down here. The green is the basically where it's secreted from. So it's secreted from this cell here, and it's going to travel in the bloodstream, so throughout the body. And then this here, these are little receptors where my pointer is. So those are little receptors that are going to attach this hormone, and now it can cause a reaction in the target cell. The next pathway is paracrine. So this is when we have cell-to-cell or cell-to-receptor communication. So secretion of a cell to a target cell that's just located nearby. And we can see this. So these cells are located next to each other or very close in proximity. And so here is the cell that released it, and then it attaches to a nearby target cell. Some examples of paracrine release. And I think we'll look at that on some few slides. Adipocytes, so fat cells, release leptin to alter metabolism of nearby fat cells. So when the fat stores are high, more leptin is released to regulate energy balance and then also to inhibit hunger. And then another hormone that uses the paracrine release is white blood cells. They release something called cytokines, which are proteins that affect the action of other nearby cells. And then next is autocrine. So auto means self. So it basically means to activate itself. The cell secretes a hormone that attaches to the receptors on the outside of the same cell. So here, this cell released it, and then that hormone goes back and activates the same cell that it came from. So examples of this, some muscle fibers have a release kind of response like this. So mechanical stimuli to muscle fibers via force production will release hormones that act on the muscle fibers that released it to stimulate protein synthesis and then eventually hypertrophy. So these are our three hormone release patterns. And again, like I said, I think I have examples here. So examples of autocrine signaling. So autocrine meaning self. So we just gave the example of muscle fibers. When they're activated, that mechanical stimuli of force production releases the hormone called IGF-1. So IGF stands for insulin-like growth factor. And we have a few slides dedicated to this hormone that we'll discuss later. But IGF basically binds to the receptor on the same fiber that released it. It has anabolic action, so it helps to stimulate protein synthesis. Paracrine signaling, so we said released and then travels or acts on a nearby cell, so a cell that's located close to the other cell. One is neurohormones, so gonadotropin releasing hormone. We actually don't really talk about this one much. It's one of the sex hormones. Neurotransmitters, so norepinephrine has paracrine signaling. I already mentioned the adipocytes. They release leptin, which helps to affect nearby fat cells. And then I also talked about white blood cells, how they release cytokines, which are proteins that affect actions of other cells as well. So these are just some examples of autocrine and paracrine. What's actually more common is the endocrine release, so when a hormone travels throughout the bloodstream. Now let's discuss how do hormones interact with receptors. So we probably already know slightly what receptors are. Receptors are basically how a hormone attaches to a target cell. It attaches to its own specific receptor. So remember when we talked a long time ago about a process called the lock and key fashion. We mentioned that certain structures or hormones can only bind with its specific receptor to exert its action. Basically, that one key only fits into a certain lock. And that's how hormones and receptors act. So there's only one specific hormone that can bind a cell or bind to a receptor. Let's talk about what the magnitude of effect depends on. And this is talking about if we have enough of a certain hormone, what will determine its effect. So magnitude meaning greatness of the effect. It depends on hormone concentration. So how much of the hormone is present in the blood? So do we have a high concentration of this hormone or a very low concentration of the hormone? Another factor that influences this is number of receptors. So number of receptors on the cell. We'll talk about with testosterone how the more trained we are, so if we lift weights, we actually have more testosterone receptors, especially males. So more testosterone receptors, and that's just to bind the testosterone hormone. And so the greater number of receptors on the cell equals or results in an increased likelihood that we're going to have more binding of that hormone. So we're going to have a greater effect. And then another is the affinity of the receptor for the hormone. So sometimes we have an increased affinity or a decreased affinity. If there's an increased affinity, it means attractiveness. So if the receptor is more likely to bind a hormone, we have an increased affinity. But sometimes there are certain cases or instances where we have a decreased affinity of that receptor for the hormone, like drugs, medications, alcohol use can sometimes decrease receptor affinity, so it's less likely to bind a hormone. Also being sick or being ill, the hormone may be less likely to bind. So these three factors determine the magnitude of the effect. Two other terms let's mention is downregulation versus upregulation. So you can see that downregulation is decrease in receptor number in response to a high concentration of a hormone. So if the hormone concentration is too high, that receptor number decreases or they become less active, because if we have a really high concentration of a hormone, if the receptors are able to bind that high concentration of hormones, then that may cause an adverse effect, and that's not something we want. So we have decreased receptor number due to the hormone concentration being too high. That just helps keep us at homeostasis to where we don't have a huge effect from a single hormone. And then upregulation would be an increase in receptor number because there was a low concentration of the hormone. So if there's a low concentration of the hormone, it means that we actually want an increase in the binding to have a balance of that hormone, keeping us at homeostasis. So the receptor number will increase to bind all the available hormones in that low concentration. How does exercise affect these actions? So I have kind of a quote from a study that looked at 1 to 2 heavy resistance training sessions increases the number of testosterone receptors. That helps to activate repair mechanisms to stimulate hypertrophy and then changes in strength. But if the stress is too great, catabolic actions can exceed anabolic actions. So the inability of binding or receptor downregulation. Exercise increases blood hormone concentration and then also increases the probability of receptor binding. This picture here just shows hormone receptor interactions. So we've looked at a similar kind of lock and key example before. So let's look at the red. We see an allstric binding site. So chemicals that interact here. So these chemicals just help this hormone, let's say, to bind correctly. So we see hormone A is our key for receptor A lock. And then we see hormone B will attach here to the surface receptor B. And then we see hormone C can only fit into that receptor C, into that lock there. But here, This hormone has the potential to bind right there. You can see that it would fit. So that's called cross-reactivity, when a hormone that doesn't have the exact same structure as another hormone, but it can still bind to its receptor. So you can see that this hormone C will fit perfectly into receptor C. You can see this hormone up here can still fit in that tiny spot right there. So cross-reactivity, and now it prevents this hormone C from binding to where it's supposed to. And then something else to point out, we'll talk about binding proteins. So hormones can extend their half-life in the bloodstream by binding to a protein. And that's how it can travel. So most hormones have a binding protein, so they can bind onto it, like you see right here, and it can travel throughout the body. But yeah, let's move on. Let's keep talking about receptor interaction. So we said we have two types of hormones. Well, we actually have three, but the two main types are peptides and steroids. So we're going to talk about peptide receptors, and then we're going to talk about steroid receptors. So we see up here, peptide receptors is what we're talking about first. So we already know what a receptor is. It mediates basically the hormone signal to the DNA, because the hormone cannot basically give off its signal unless it interacts with the DNA, because the DNA, that's where all the genetic material is. And so in order for the hormone to exert its signal, it has to get into the DNA and have a gene activation process. So how do peptides interact with the DNA? So I want you to look at this picture down here. This is, the green is our peptide hormone, and this is its receptor. So it can bind onto this receptor, but what I want you to look at is this receptor sits on the outside of this cell. Because if you look here, this is the cytoplasm, and then this is the nucleus. Out here, we're still kind of in the bloodstream. So this hormone cannot cross into the cytoplasm. It cannot travel. It's not lipophilic. So lipophilic means it can travel across a lipid-based membrane. This hormone cannot. Peptides cannot do that. So they attach to the receptor, which is on the outside, kind of in the extracellular domain. It's basically not a fat-soluble polypeptide, and it cannot pass freely through the plasma membrane, but it binds to the receptor on the surface of the cytoplasm. And then it uses a secondary messenger to generate a signal in the cell. And we can see that secondary messenger right here. So the receptor will change shape as the hormone binds. So receptor changes shape, and then we have our secondary messenger. So this is another chemical messenger that now helps to carry that signal into the nucleus, where it can act with the DNA and cause some sort of gene activation. So what hormones are peptides? We already mentioned insulin. Another one we'll talk about is growth hormone is a peptide. And then epinephrine, norepinephrine, and serotonin are also made of amino acids, and they have a very similar effect here. So peptide receptors, what you need to know is they rely on a secondary messenger to get that signal from the hormone into the DNA. But steroids are different. So steroids are lipophilic. So if they're lipophilic, they can readily diffuse across a lipid-based membrane of that target cell. And then they can bind to the receptor in the cytoplasm to form an active receptor hormone binding to interact with the DNA directly. Steroids basically don't rely on another messenger. The receptor is located within the cell, not in the plasma, and they contain a specific sequence of DNA, basically something called a hormone response element. So they all share a similar structure and functional characteristics in DNA binding, which results in regulatory protein involvement. So just to kind of sum that up, we see our steroid, our yellow guy out here. It diffuses across the plasma or the lipid-based membrane into the cytoplasm. So it binds to its receptor, and then the receptor will then pass that hormone across the nucleus, and then it can then interact directly with the DNA. Some steroids that use this, cortisol, and testosterone are both fat soluble, and they're both steroid hormones. All right, so let's look at a picture of this one more time of how this steroid action works. All right, so we see our carrier protein at the bottom. So steroid is transported in the plasma bound to a carrier protein. So the blue triangle, that's our hormone that we're looking at. So we're looking at our blue triangle, and then I already mentioned a binding protein or a carrier protein. Basically kind of carries the hormone throughout the bloodstream, helps to extend its half-life. Okay, so steroid is transported. And then you see that steroid, the blue triangle will diffuse across that plasma membrane. So it releases from the binding protein. We already said it's lipophilic, so it can travel through this target cell. So now we're in the cytoplasm. So all this purple area is the cytoplasm of the cell. So now we can see it's going to attach onto its receptor, so our orange triangle there. And then that receptor allows that hormone to then transport into the nucleus. So as it transports in the nucleus, we're then going to have interaction with the DNA. So something called mRNA will help to stimulate protein synthesis. RNA we know is ribonucleic acid. It's a molecule that carries the message contained within the DNA to the ribosomes. So ribosomes are where the proteins are produced. So mRNA is important because the ribosomes cannot reach the DNA inside our cell nucleus, which is the location inside the cell where the DNA is located. So basically, protein synthesis is not possible without this mRNA. You don't have to know a ton about this process here. This process is just to show you how steroid action works. Just know that as that receptor interacts with the DNA, it will also stimulate mRNA to cause a reactional protein synthesis, which we see down here at the bottom. So let's look. So this is basically steroid action. All you need to know is that the steroid can diffuse through the cytoplasm, so the lipid-based membrane, and it has a direct interaction with the DNA, which in this case, or this example, we have protein synthesis. This next picture here shows secondary messenger system. So now we have peptide hormones. So peptide hormones we know cannot diffuse across the plasma-based membrane. So what happens in this process here? All right, so we look in our picture at the very top and we see the lock-in key of the hormone and the receptor. So the hormone binds to receptor. You see it's on the outside of the plasma membrane. So we're not inside the cell. We're kind of on the outside of that cell wall. So what happens when the hormone and the receptor bind together? That activates a G protein that we see here. So what is the G protein? We have a link between a hormone-receptor interaction. G proteins are basically a family of proteins that act as molecular switches inside the cell. They're basically involved in transmitting the signal from a variety of stimuli outside a cell to its interior. So basically this link here that basically switches on kind of and allows for signal activation. What happens next? The G protein, as we follow over here, activates adenylate cyclase. So here's our adenylate cyclase. We see first let's talk about what is this structure. It's an enzyme with key regulatory roles in essentially all cells. It's the most polyphyletic enzyme. Six distinct classes have been described. They basically just kind of help to interact and cause ATP production. Another way to describe adenylate cyclase, it's kind of a catalyst. So it's an enzyme that catalyzes ATP into cAMP, and we see cyclic AMP right here. So that's the role. It's just an enzyme that facilitates ATP into cyclic AMP. So now let's talk about what is cyclic AMP. So I have this statement right here. Cyclic AMP activates enzymes that bring about cellular responses. And we can see we cause a cellular response here at the bottom. But let's talk a little bit more about it. So cyclic AMP stands for cyclic adenosine monophosphate. So we know with ATP, it's triphosphate, mono meaning one. So we have adenosine molecule plus one phosphate, which is a second, so the AMP is a secondary messenger that's important in a lot of biological process. Cyclic AMP has associated kinases with that, and we can see a protein kinase here is inactive, but it causes this protein kinase to become active. So these kinases function in several biochemical processes, including the regulation of glycogen, sugar, and lipid metabolism. So back to this protein kinase, what's the role of this? It's a kinase enzyme that modifies other proteins by chemically adding phosphate groups to them. So it's basically phosphorylation that usually results in a functional change of the target protein by changing enzyme activity, cellular location, and association with other cells. So those are just kind of all the definitions of these. All you have to know from this slide, you're probably like overwhelmed with all this information. You've never heard of these words before, but just know we are talking about peptide hormones. They're polypeptide amino acids. So just know that how do they interact with the DNA? They use a secondary messenger. So the secondary messenger in this case is a G protein, and it's going to go through a series of reactions that eventually produces a cellular response and a change within the DNA. So that's really kind of all you have to know about this slide here.
There are two main mechanisms for which a hormone can affect a target cell. The first is by a non-steroid action. This mechanism is employed by an amino acid hormone or a peptide hormone. These hormones cannot freely cross a membrane on a target cell, so it first acts by binding a cell surface receptor. There is then an intracellular signaling cascade that occurs in order for the desired effect to take place. There are proteins on the intracellular side of the target cell that are associated with the receptor. Most often, this involves what's called a G protein. This G protein is found next to an enzyme that converts ATP to a molecule called cyclic AMP, or CAMP. This is something that makes this mechanism unique. Cyclic AMP is called a second messenger because it signals a cascade of events that eventually change the enzymatic activity in a cell to cause the target cell response. Examples of hormones that use the cyclic AMP second messenger system include ACTH, calcitonin, epinephrine, glucagon, parathyroid hormone, and ADH. The second mechanism is the steroid action. Because lipid derivative or steroid hormones are lipophilic, they can freely move through the membrane and into the cell. From there, they can either bind to a receptor within the cell, or they can move freely into the nucleus, where the hormone, or hormone complex, causes a change in gene activity. This then increases transcription and mRNA production, which then leads to an increase in protein production within the cell. Examples of hormones that exert effects via steroid hormone mechanisms are testosterone, estrogen, progesterone, aldosterone, and calcitriol.
All right, so hope that helped explain each of these actions a little bit better. All right, so next let's talk about feedback systems. We've already talked about feedback loops in chapter 5. We talked about positive and negative. We're basically talking about the same thing, except now related to hormone action. So negative feedback, well, first let me start with feedback systems. The control of hormone secretion is affected by different feedback systems, which in part control the amount of hormone that's released by a gland. We have negative and we have positive. So negative feedback, we can see that secreted hormone acts to decrease its secretion from a gland. This process is used by most hormones. The hormone that is formed stimulates the gland or the structure that secreted the substance to diminish the amount of secretion or physiological function stimulated by that hormone. So if a hormone is too high in the blood, that hormone will act on the gland that secreted it to inhibit its production. Two examples of this: first, testosterone, and then second is cortisol uses negative feedback. So when testosterone is too high in the blood, the body recognizes that. So testosterone will go back and act on the gland that released it to inhibit its production. Next, let's talk about positive feedback. So now we have a hormone that acts to increase its secretion. This is less common, but the hormone stimulates the gland that secreted it to increase the amount of secretion. Oxytocin uses this feedback system. It's released from the posterior pituitary gland. I think we mentioned it before. It's released during childbirth to strengthen the labor contractions. And then it also stimulates the mammary glands to increase milk production, which influences breastfeeding. But yeah, and then let's talk about the next one. So next one is not positive or negative, but it basically requires multiple hormones to have a multiple feedback influence system. So it's the redundant regulation of a vital process. So these multiple feedback influences, they're signals not based on feedback from one hormone, but from several systems of hormones. The example is multiple anabolic hormones. So testosterone, growth hormone, and then IGF, which is insulin-like growth factor. Those are going to increase to create an anabolic environment to stimulate protein synthesis and hypertrophy of muscle cells. So with the multiple feedback influences, just know it's multiple hormones acting together to cause a process or to keep regulating a certain process in the body. So just know these loops and hormonal influences are important for exercise training, adaptations, and recovery. So what you have to know is kind of just the definition of the three systems that you see here, and then possibly an example of each one. So the first one we said was testosterone and cortisol. Second one is oxytocin. And then the last one is just the anabolic hormones in the body: testosterone, growth hormone, and IGF. But that's all we have for part one. So I'm going to stop this video here, and the next video will be